Hello friends, today we are going to learn a very important and interesting topic in microbiology, that is gram staining technique. Introduction. Gram staining is a differential staining technique used to classify bacteria. It is developed by Hans Christian Gram in 1884. Gram-positive bacteria classify two groups. They are gram-positive and gram-negative bacteria based on the cell wall structure. Now, let's see the principle. Gram-positive bacteria have thick layer in the cell wall called peptidoglycan. This layer holds the primary stain, crystal violet, very well, so these bacteria appear purple under the microscope. Gram-negative bacteria have thinner peptidoglycan layer and an outer membrane. They lose the primary stain and take up the counter stain, safranine, so they appear in pink red color. Materials required. We need some basic materials: a clean glass slide, inoculating loop, Bunsen burner, staining reagents, that is crystal violet, grams iodine, alcohol, safranine. Finally, we need a microscope to see the results. Now let's see how to gram staining step by step. Step 1 is smear preparation. Take a clean slide, add a drop of water and mix a little bit of bacterial culture to make thin smear. Let hair dry completely. Step 2 is heat fixing. Passing it through a flame 2 or 3 times. This helps the bacteria stick to the slide. Here, staining methods. 4 major steps. One is primary stain, that is act as a crystal violet. Iodine act as a mordant. Decolorizer, alcohol. Safranine is a counter stain. Then step 3 is a primary stain. Flute the smear with crystal violet for 1 minute. This is our primary stain. Then gently rinse with water. Step 4 is mordant. Grams iodine for 1 minute. This acts as mordant, forming a complex with the crystal violet inside the cell wall. Step 5 is decolorization with alcohol or acetone for 15 to 30 seconds and rinse immediately. This removes the stains from gram-negative bacteria but not from gram-positive ones. Then step 6 is counter stain with safranine for 1 minute. Rinse gently and let the slide dry. Now gram-positive bacteria will appear purple color and gram-negative bacteria appear pink color. This is the observation. Observe under 40x and oil immersion under X microscope. Gram-positive result is purple, example Staphylococcus aureus. Gram-negative pink color and red color, example is E. coli. This is gram-positive cocci. This is gram-positive bacilli. This is gram-negative cocci. This is gram-negative bacilli. Finally, the conclusion. Gram staining helps identify and classify bacteria quickly. Provides clues about bacterial type for diagnosis. Basic and essential technique in microbiology labs. That's it. This is how we do gram staining in microbiology. Thank you.